Yep.
Make this happen. Great. Well, thanks for everyone. Thanks to everyone for being here that way around. Gosh, not enough coffee yet. Lovely to see you all, especially as we just discussed, today is a holiday in some countries. So it's lovely to see you nonetheless. Now, today, I thought I'd take advantage of Victor hosting this for the last month, and I actually present a paper myself because it's always easier presenting when you're not doing all the tech live stream on the side. So let me share my screen with you. Can you see it? Great. Okay, so as you can, hang on, let me copy the link so you all have the link to the paper as well. Here you go. Victor, if you could share that on YouTube as well, that'd be great. Thank you. So I came across this paper last week and I thought it was absolutely exciting. So that's why I'm presenting it to you, and I'm telling you why I thought it was exciting. So the paper offers a unified 3D map of the human brain, combining histology measures, immunohistochemistry, and 70 MRI measures. The whole data is available for 2 full brains from 2 women aged 75 and 95 with no neurological disorder. And all the data was co-registered to a sub-millimeter space of 200 micron within a sampling interval of 600 to 1000 micron for the histology data, I believe. Now, what this allows you to do is to look at the neurons, the glia cell bodies, the interneurons, and the fibers in the same sample across different modalities, and then they brought it all to the MRI space, so you can plot your own data over it if you like, and everything is made open access. So that includes all the data and the scripts. Now, why is this of interest? It's of interest because we don't really map the subcortical structures of the brain a lot, partially because some of them are just too small for MRI to see them. And then usually bringing together histology and MRI is not A, easy, B, not many people cross those interdisciplinary boundaries. So this work allows, together with others who've done similar work, to map an atlas of the small structures of the brain, so the subcortical nuclei. We can map the cortical laminar structures across modalities. They also show a vascular map of the brain, a full map that I'm happy to discuss a little bit more in detail later on. And then they also use the data to automatically parcellate the subcortical structures of the brain. So let's have a quick look at what the results look like. There's also a video in the supplementary material where they fly through the different modalities at the same time. So if you're interested in that, go and watch that on the supplementary material. So this is quite a dense figure, and as you can see, I was making my notes to try and get through it. But basically what we have here is an overview of the 3 MRI contrasts they used, which is the longitudinal relaxation R1, the transverse relaxation R2 star, which is sensitive to iron, and then a proton density map. Then we have a blockface histology image. Is anyone in this room who knows histology, done histology? No? Gosh, you have to do with my... Oh, there's it there. Can you explain what a blockface is?
Nope.
Okay. In that case, we're going to go with my...
I know the kind of process. It's the co-registration process, the way histology is co-registered to MRI.
That's what I got as well. So it's co-registered along the Z-axis for volume reconstruction from a series of assembled slices. You can totally see my notes here, right there in the figure.
It's not a difficult process.
And that allows you that blockface configuration, which apparently has limited tissue deformation. That's as far as my understanding goes about blockface. But I'm making this point because that's the important image they use to co-register everything too. So that's our reference image. Hence, I spent some time trying to understand what it is. Now, why did they use that? They used it to avoid the banana effect, which I thought was a hilarious term. And that basically means when you do the process, you sometimes have that problem that the image is bent and therefore distorted, which apparently if you use the block face is less of an issue. And then we have two more histology stains, the Nisser stain for cell bodies and the silver stain for fibers or the white matter. And then there is 3 immunohistochemistry stains that allow us to look at the interneurons. So quite a dense data set, as you can see, but quite beautiful images, and you can nicely see how the subcortical structures are coming out with different contrasts in different ways. Now, another thing they showed, as I said, was the vasculature. So on the left here, you just have the 3D surface rendering of the block face, the R1 and the stains. And then here on panel B, you have the vasculature image of the brain, which is a whole brain picture of the vessels, which I think is a beautiful picture. I'm struggling to see the small and large vessels, to be honest. And I also have tons of questions about the variability, because we know the brain is variable, we know the gray matter is variable, the white matter is variable, but that variability is nothing compared to the variability of the vasculature between people. So that could be quite an interesting means to investigate that. Then here in panel C, they also show the automatic parcellation, but they made the point right here that still manual delineation remains the gold standard. And we, especially in this group here, had this discussion many, many times. So I just wanted to link the automatic image with the note that manual delineation is still the gold standard when it comes to neuroanatomical work. And then panel D shows us the thalamus using different stains. And the point here is that with different stains, the boundaries and the shape of the thalamus turns out slightly different. So it's important to know the question that you're asking in terms of the data that you are then using. Now, as I said, everything they made open access. So here you have the overview of the staining, the 3D reconstructions, the MRI co-registration, the automatic parcellations, and all the Python scripts they used. So this should be plug and play, so to say, if you want to use the data. Then they also reconstructed the tensor shape here. Now, the original MRI images were acquired at an isotropic resolution of 400 micron across the 3 contrasts, but obviously this is post-mortem, so you can scan for a much longer time at a higher resolution as well. And we can see, we can zoom in slightly. Here we go, beautiful tensor maps where they looked at the white matter, but then we know the limitations of the tensor. So while this is a fantastic image and beautiful, how it really compares to in vivo tractography results needs to be determined. And then again, a density map of the blood vessels down here at 20 micron sections that were realigned to 200 microns in the 3D. And we can see that the brain is densely packed with blood vessels. But again, untrained eye, but I don't see the difference between the large and the small vessels, which coming from a stroke background obviously makes a difference. And then we are diving into the methods, which I'm not gonna spend too much time discussing, because we can either dig it out together, or for those of you who are really interested in how it works, you can go back and read them. But as I said, they use the blockface image as the reference. Here we go. And they did a forward and a backward co-registration with the neighboring stains, and they went 3 back and 3 forward, registered it to the blockface in 3 iterations, and then also registered the MRI to the histology image using ants, which we're very familiar with. That is pretty much, oh yeah, the banana effect. I may have started procrastinating a little bit towards the end. I read it very late. That's pretty much it. I thought it was quite an exciting paper and offers a lot of data to use and play around with. And I'm happy to discuss it a little bit. Any questions? Comments? Some of you need to have a question, because I can't ask myself questions. Victor, here we go.
Yeah, thank you for the presentation. Really interesting article. I have 2 kind of small questions. One is about the quite obvious artifact that we can see often on the images, because here, for example, we can see it very well. This one, okay, I can see that all the lines that we can see a lot. What are they exactly? Because I expect them on things that are sliced independently, and I guess this is the case. But for the MRI ones, I think it's kind of weird.
So bear in mind, these are 2 postmortem brains.
Sure.
So you're very likely to have artifact in postmortem brains. And they also mentioned that one of the brains had more artifacts because of the pre-processing steps that were introduced. Also, they, hang on, that wasn't a methods. They got the brains following a whole body perfusion, which is also not necessarily the perfect way to preserve the brain in the first place. So if you want to have the most stunning brains, I guess you take the brain out and preserve the brain separate from the rest of the body. But due to the way that you often get access to postmortem specimens, this is a common way of tapping into the donations and getting those specimens for brain analysis. But obviously it's not optimal for the brain, and then that introduces artifacts in the process.
Okay, thank you. I mean, you do what you can with what you have, right? So
exactly. And they still manage to do a fantastic job.
That is impressive. That's true. Yeah. And the other question is more about my ignorance about this specific stuff, about the vasculature of the brain. Can you go up a little bit, please?
Oh, this one
perfect.
or the one?
No, this one, perfect. I can see that it's a cross-section every time. It's not like a 3D view of the brain here. So I can see that there is a lot of blood vessels in the white matter, and I was expecting more vasculature in the gray matter, actually. So do you have any knowledge about that, or is it an artifact, or is it normal? I don't know. Personally, it's I discovered this.
I mean, in brackets, I'm not an expert on vasculature, but it's definitely something that we do not study when we study neuroanatomy of the brain. Definitely not enough. We all heard about the circle of Willis, which are those huge vessels at the ventral surface of the brain, but we never really look at the small vessels and arteries in the brain. Now, it's not an artifact. There is small vessels everywhere in the brain. So this is very convincing, I would say, especially with working with stroke for 16 years. I've seen a lot of patients who had small or so-called lacuna strokes that are subcortical, and they usually, not always, but usually don't cause tremendous physical and cognitive symptoms because you block a small vessel in the brain, and then the tissue behind it is damaged or dies away. But that usually can be compensated. Whereas if you have a stroke to a large vessel, like your middle cerebral artery segments, then that damages a very large subcortical and cortical part of the brain. And then you clearly present with clinical symptoms. So that's why I made the point earlier that I don't really see any difference here between large and small vessels. That would be interesting to me personally. But I'm sure you can probably tease that information out.
Yeah, because as someone who works mostly on MRI and fMRI especially, I'm used to have most of the signal from the hemodynamic signal in the gray matter. So where there is consumption of oxygen and the response by the hemodynamic response is usually mostly in the gray matter. So I would have expected more vasculature in the gray matter than in the white matter. But because, as you said, we don't have the difference between the very small vessels and the bigger vessels, I don't know here how it's, if it's a reason for this kind of distribution. But yeah, we are not expert here. Maybe there is an expert in the room who might enlighten us, but we'll see. No? Yes?
So cortex versus subcortical. A is not a clear differentiation like that. It's not like the dichotomy of you either look at the cortex or you look at the subcortex, subcortical structures. They go together, right? They're densely connected. They work in integration. But also when we look at strokes, there's a discrepancy between what we know from the literature, which is the cortical big lesions compared to the lacuna small lesions. And the difference is that the cortical lesions account for roughly 15% of the strokes, yet the literature is disproportionately talking about cortical strokes. So that's something we probably also see when we look at fMRI, that there's this discrepancy of what we're looking at, what we focus on, and the information the brain has to offer.
Sure, thank you. I think Hervé has a question.
Yes.
Yes, I have a question. Thank you for the presentation, Steph. So it's a really it's a basic, naive question about this study. They talk about the post-mortem interval below 24 hours. What does that mean exactly?
I take that to mean that they started the process and took the specimens within 24 hours of the person dying.
Between the death and the MRI and everything? Okay.
Yeah.
Because it's fast.
That, yeah, if that is it, then this is very, very fast.
Wow. So that's it.
But that was my interpretation of it. You're right, it's not entirely
Maybe
right.
it's not like the imaging, if not the perfusion. No?
Ah.
That's why I'm saying it's not entirely clear if they got the brains within 24 hours or if they, yeah.
Because I think otherwise it's too fast. Usually you have to wait before like some hours when the person died, like some hours for like certify the death. Also maybe it's
You're
like
right.
24 hours before the perfusion, and then one is the brain is perfused. Yeah, they can. It's like.
So as I said, to me, this paragraph here is not entirely clear what the order was. I know that when I worked with whole body perfusion specimens, it took a couple of days minimum for me to get access to the specimen. But then I guess it depends on how the study was set up. If this is all discussed before, maybe even the people knew before and said, yes, that's fine, take it. It all depends. But to me, the way it's written here is not entirely clear what the timing was. But I would read this as they obtained the 2 specimens within 24 hours following the perfusion. So that must have happened very, very quickly.
Okay. Thanks.
You're welcome. That was a quick one. Oh, Cesare has a question.
Yeah, hi. Thank you for the presentation. Really nice, really beautiful. I didn't get, I don't know if you said it maybe, but I didn't get if the 200 micrometers sections, we were talking about thickness, right, of the sections?
Yeah, so they specified thickness and slices. So to be honest, I read this late at night yesterday and I got lost a bit in the numbers because there is the sampling interval between 600 and 1,000. There's the co-registration to 200 micron.
No, I was just
800 microscopy sections. So it was too many numbers for my brain in the middle of the night. But I remember they specified the thickness. Hang on, can I just find it? Thickness. They specified it somewhere. Well, it's not helping if you made the entire paper yellow and the search function is yellow. Oh, there we go. Not the part I was looking for. Can't quickly find it, but I know it's in the paper.
okay. No, because I was curious to know, like, to do blockface, you have to cut the sections, the coronal sections. So I was wondering how big those cuts were. That's it. And I wasn't sure about the 200 micrometers thickness because I wanted to understand if it was like, I don't know, the area of the blockface or something else. I'll read it and I'll discover it myself.
So the 200 micron is the final data, the co-registered data. The section should be a lot thinner. But I have to be honest because I'm no expert in histology. I just took a mental note and glanced over it, but it was somewhere in the paper. So if you have a look, you'll definitely find it.
Okay. Thank you.
Well, great. If there's no further questions, we actually finish on time this week. Thanks for joining us this morning, live here on Zoom and over on YouTube. And we'll see you next Monday for the next Neuroccino. Any volunteers for next week, just let me know when you come across some exciting science that you want to present. As I said, everyone is welcome. We take papers, we take books, we take book chapters. We also recently expanded to YouTube videos. So anything that takes your fancy, you can come and present to us. Just let me know throughout the week and I reserved a spot for you. With that, I wish you an exciting week in science and I see you next Monday. Bye.
Thank you.
Bye.